So I've been coming to these meetings since the mid-1980s, and one thing I think that's been consistent from almost every meeting I've been at is that viruses get a pretty bad press on the whole. They infect this, they infect that, they kill this, they kill that. And the kind of, my pet noir animal virus is foot-and-mouth disease. And just to give you a couple of, for examples, in 1997, there was an outbreak in Taiwan, and that outbreak actually destroyed 90% of their pig industry. And of course, we all know about the 2001 outbreak in the UK costing 5 to 10 billion pounds, which, okay, post financial crisis and Brexit is perhaps not quite as big a sum as it used to be, but nonetheless, it's a fairly substantial amount of money to lose. But as Neil said, I'm going to try and persuade you that there is almost a silver lining to this cloud, in that the study of the way that this virus controls its protein biogenesis led to these amazing uses by people a lot smarter than I in biomedicine and biotechnology. So I want to kick off by acknowledging the people that made this happen. So Gary Luke has been in my laboratory for just about 20 years now, and he's worked in every aspect that I'll be talking about today. He's probably forgotten more about 2&8 than I'll ever know. Michelle McNabb, Nay Donnelly, from the papers that arose from our lab, she was a PhD student with me and then went on to do a postdoc, and kind of was very influential in determining how the mechanism of this thing works. Lorraine Hughes in the publications, she did a huge amount on 2A-like sequences from different viruses. Pablo De Filippi was involved in the developing this system for gene therapy applications, and Claire and Valerie, well, again, we're involved in something that I'll only be able to touch on tangentially, which is cellular 2A-like sequences. And these other people here also have contributed to the story, and not forgetting the other people in the lab working on foot-and-mouth disease virus replicon systems, but their work also feeds into this kind of whole area. Collaborators, University of St Andrews and latterly Birmingham, now retired. Professor David Garney was an organic chemist at St Andrews. And then at the University of Dundee, my wife, Claire Halpin, who's in the audience, and Claire in her laboratory at AstraZeneca first showed how this thing could work in transgenic plants. And then critically, a fantastic collaboration with Jeremy Brown at the University of Newcastle. And I was giving a talk at Edinburgh, and then you get wheeled around to speak to people. I met Jeremy, we struck up a collaboration, and virtually all of the work that I'll be talking about the mechanism of how this thing works, all that data arose from Jeremy's laboratory. And there are actually data presented here today from Pippa and Man about the mechanism of the way this thing works. And of course, not to forget the funders. The BBSRC has supported this work for many years, and then the Wellcome Trust and the MRC supported it for development of gene therapy applications. Right, so Michael Rossman covered this pretty well about the structure of picornaviruses, but I should point out that these things have got a positive mRNA sense single-stranded RNA genome of about 7,500 to 9,000 bases long, and the naked RNA is infectious. What that means is that the virus RNA, when introduced into the cytoplasm, it can function as a messenger RNA. And some headline viruses you may recognize, things like poliovirus, EVA71, which is human hand, foot, and mouth disease virus, and the rhinoviruses, causing about 50% of common colds, hepatitis A, and then foot and mouth disease virus. But these viruses affect a range of mammals, birds, reptiles, and fish. So going back to the 1980s, most of the research in this area was being conducted on human enteroviruses and human rhinoviruses. And as you can see, the organization of this viral genome very closely resembles that of a eukaryotic messenger RNA. The only differences really are at this end here, where you don't have a 7-methyl G cap. You have a short oligopeptide, VPG, which is this protein here in the polyprotein, covalently bound to the RNA, and then you have one of these internal ribosome entry sites, and it's actually there that the initiation of the translation of this polyprotein actually occurs. So complete genome sequences, reverse genetics, various experiments showed that, in fact, the way that this poliovirus genome encoded and was able to produce these multiple different proteins from a single open reading frame was via the agency of two virus-encoded proteinases. So the 2A proteinase here cleaves at its own N-terminus to separate the capsid proteins domain, the P1, away from P2, and then this other virus-encoded proteinase here cleaves between 2C and 3A to separate these domains. So these are what are referred to as primary processing products. You never see the polyprotein within infected cells. These are the things that you see, and then this 3C proteinase goes on to cleave these precursor forms to the mature products. So 2A and 3C, they're mimics of well-characterized host cell proteinases, except that the active site nucleophile of these proteinases is cysteine rather than serine, as would be the case in trypsin and chymotrypsin. So 60 copies of these things plus a copy of RNA makes an infectious particle. So, going back to the 1980s again, it was thought that a paradigm for protein biogenesis was emerging for all picornaviruses. And it was thought that all picornavirus polyproteins were processed only by virus-encoded proteinases. There were no host cell proteinases involved whatsoever, and that this primary processing was a co-translational cleavage in cis, mediated by these two proteinases, and then one had secondary processing with post-translational cleavages, both in cis and in trans, to give rise to these mature processing products. However, when the first genome sequences of foot and mouth disease virus and a cardiovirus were coming out, it was quite clear that there was something different in these particular genera of picornaviruses. So, in foot and mouth disease virus, where you have a 2A proteinase in this position in the enterovirus polyprotein, in 2A, it's only 18 amino acids long. And at the N-terminus of the polyprotein, you have another proteinase here, again, a mimic of a host cell proteinase, at this time thiol proteinases. However, when you look at the primary co-translational cleavage products from this, what you find is L cleaving at its own C-terminus, and then this primary co-translational cleavage in cis here, and then 3C cleaving at that position. So the thing that got me interested was, what on earth is doing this primary co-translational cleavage in that position? So zooming in on the 2A sequence itself, here's the capsid protein 1D upstream of 2A, and the actual length of 2A is defined by this cleavage event between this glycine and the proline, and then 2A is trimmed away from capsid protein 1D by the 3C protease. I should point out that this proline here that derives from the N-terminus of protein 2B is absolutely essential for this cleavage. So hereafter, when I refer to 2A, what I actually am really referring to is that sequence plus this proline. It's absolutely essential in the mechanism. So, how could a 2A of 18 amino acids bring about this cleavage? I should say that whilst I was at Pirbright, I made a whole series of deletions of sequences upstream of 2A and downstream of 2A, because at that time I thought maybe 2A could be the business end of a larger form. And all those data showed that you did not need any of the foot and mouth sequences upstream or downstream of 2A. So the next thing to do was to actually insert 2A into the middle of a completely artificial polyprotein system. So now we've eliminated all foot and mouth sequences. So the analysis here is using an in vitro transcription translation system. So this is a cell-free translation system. You supplement it with amino acids and radiolabeled methionine. So when you look at de novo protein synthesis, you're only seeing these products here. So in this single open reading frame, we've removed the stop codon from the GFP. You get just a single band here. However, when you insert 2A in frame now, you get this GUS product and the GFP2A. And this is where Michelle Donnelly comes into the story, because she kept saying, when I went up to St Andrews, the department got a phosphorimeter as part of the dowry, as it were, and that enabled us to actually accurately count the radioactivity in these bands. And the problem was that if you look at GFP, it's about 27 kilodaltons in length, but it only has 6 methionines, whereas GUS, 70 kilodaltons, has 12 methionines. And I'll hope you agree that that band is actually more intense than that band. So I told Michelle, well, maybe it's just RNA degradation or protein degradation of the GUS. And she did a lot of experiments and showed that neither of these things were responsible. So we were getting a molar excess of GFP2A compared to GUS, and that was not consistent with 2A as a novel proteolytic element, nor with it acting as a substrate for some form of host cell proteinase, in which case you'd have expected a 1-to-1 molar ratio. And that's even more starkly illustrated here, where we swap the gene order around. Now we've got GUS, 2A, GFP, and again, I hope you'll agree that now we've swapped the stoichiometry of those two cleavage products. So using these in vitro translation systems, that gave us the vital clue about the mechanism. It was nothing to do with proteolysis, and then we started thinking in terms of its effect upon translation, and that this imbalance must arise from differential levels of synthesis. We appear to be making more of whatever was upstream of 2A than was downstream of 2A. So you look at the sequence, you think you're going to get one of those, but in fact what you get is a couple of those instead. And this now we know fits into this notion of translational recoding, that 2A is just one type of mechanism that a lot of these viruses use to reprogram the translational apparatus. And the collaboration with David Garney led to a lot of dynamic molecular modeling and natural sequence variation together with site-directed mutagenesis allowed us to come up with this model. We thought this was going to be an alpha helix from the dynamic molecular modeling, and site-directed mutagenesis indicated the D and the E and the NPGP were absolutely essential for the cleavage to occur. And we knew this was highly conserved. We thought this might constitute a tight turn. And more than that, this motif gave us a tool to probe databases. So, the model, how did we think it worked? So the model that we came up with, and this is what passes for a thought process on Planet Ryan, was that activity maps to this oligopeptide sequence of 20 to 30 amino acids. We know it's co and not post-translational, so it might be acting actually within the ribosome. And hold on, the ribosome exit tunnel accommodates 30 to 40 amino acids of the nascent polypeptide chain. So maybe 2 interacts with the exit tunnel of the ribosome, which might be affecting stereochemistry within the peptidyltransferase center of the ribosome. And the idea was that the peptide bond synthesis at this particular site at the C-terminus of 2A was inhibited. So now we're getting into some industrial grade arm waving. The idea would be that we've jammed the translation at this point. The peptide bond cannot be made. The prolyl tRNA leaves the system. Somehow or other, we've got hydrolysis of this glycyl tRNA ester linkage, which then releases the nascent polypeptide made up until that point. And then somehow or other, translation reinitiates. So here we'd have to propose that the prolyl tRNA can re-enter the system and be translocated across, and synthesis carries on. So the idea is, literally is a game of two halves. You make one bit, you stop, and then you make the other bit as a discrete entity. So just to summarize that here, so here's 2A interacting with the exit tunnel of the ribosome. Here's the nascent polypeptide. Here's our glycyl tRNA in the P site, our prolyl tRNA in the A site. That system is jammed. because this interaction has altered the space that can be occupied by that glycyl ester linkage here, which would preclude it from attack by prolyl tRNA. And the reason why that has to be prolyl tRNA is that proline is a sterically hindered nucleophile. It's probably the worst amino acid you can try and get to add onto a growing chain. So the system is jammed at that point. We propose that the prolyl tRNA exits the system. Somehow or other, we got hydrolysis of this ester linkage to release the nascent peptide, and then prolyl tRNA re-enters back into the A site and is translocated from the A to the P site by the agency of elongation factor 2. And then you can just make the downstream sequences. Now, as I said, I met Jeremy Brown when I was giving a talk at Edinburgh. And Jeremy was working on yeast, and the idea was that we would develop a phenotypic screen for 2A-mediated cleavage in yeast. And if we could do that, we could bring the power of yeast genetics to bear to try and find out what host cell factors were involved in this cleavage mechanism. And since 2A works mid-elongation cycle, we thought we could eliminate initiation factors, we thought we could eliminate termination release factors, and obviously the ones we thought would come up from this screen would either be elongation factor 1 or 2, or maybe ribosomal RNA or ribosomal proteins that constitute the exit tunnel. So Jeremy did this and did the screen, and of course it turned out to be termination release factors. So Jeremy showed that yeast defective in release factors 1 or release factor 3, this affected this 2A-mediated cleavage. So initially it was identified by genetic screens, but Jeremy's lab went on to confirm these by biochemical analyses, and there was actually a poster here confirming even more data about that. So the idea would be that we can put a bit more meat onto this model now. So, same as before, the prolyl tRNA exits the system, but now what we think is going on is that release factor 1 comes in and performs the hydrolysis on this ester linkage, which is exactly what it does normally during termination of translation. But then, of course, we have to postulate the egress of release factor 1 and 3 from the system, which allows prolyl tRNA to come back in so that you can get continued translation. Or you terminate at that point. And the reason why that's significant is that now explains the imbalance that we saw using those in vitro translation systems, how we could make more of whatever was upstream of 2A than was downstream of 2A. So you could either reinitiate to make the downstream product or terminate at that position here. So it turns out now that we know that 2A is a member of a family of ribosome arrest peptides. There's now quite a number of these things being characterized, both from prokaryotes and from eukaryotes. And 2A does not work in prokaryotes because of these differences in the geometries of the ribosome exit tunnel between pro and eukaryotes, which confirms, adds weight to our notion of 2A interacting with the exit tunnel of the ribosome. And there's a fantastic review on that whole topic of arrest peptides in this review here. So that model that I presented about the bulk of 2A being in an alpha helical conformation, that was based on dynamic molecular modeling, but also some theoretical work about the way that tRNAs progress through the ribosome and rotate, and how that might impart a coil structure on the emerging peptide. However, again, recent data from Jeremy's laboratory has now shown that in fact that part there is in an extended conformation in the ribosome. And there was a talk given earlier on today by Pippa from his lab, and that's the kind of data that they used to finally solve that this was an extended conformation in the exit tunnel of the ribosome. So I mentioned already about this glycine and proline here, And interestingly, if you look way back in the literature, you find there are two lines of evidence to support why glycine and proline are in the position that they are. And this was early experiments done on the so-called partial reaction when translation was being characterized, and the most inefficient amino acyl adenosines were A-gly and A-pro. And when people started synthesizing analogues of puromycin, for example, the worst analogues you could possibly make were proline. And the most inefficient proline analogues, were proline and glycine. The one most powerfully inhibited by puromycin was again proline. So as you can see, there's something rather special about this glycine-proline cleavage site. So I mentioned that we've got this conserved motif at the C-terminus of 2A, D, V or I, E, X, NPGP. And then we can now use these things to probe databases. And I'll be talking mainly about 2A-like sequences in RNA viruses, but I'm going to go off-piste a little bit and just have one or two words about cellular 2As. This is some work that we've been doing recently. And we've found cellular 2As in a range of genome sequences, mainly from invertebrates. And the first class of gene that we found these things in is in non-LTR retrotransposons, which comprise about 17% of your entire genome. So characteristically, the young type of non-LTR retrotransposon has a short open reading frame 1, a long open reading frame 2, which encodes an AP endonuclease domain. This is how the element reinserts itself back into the genome in a sequence-independent manner, and then an RT domain. And what we found inspecting these genome sequences was that there was a whole class of non-LTR retrotransposons from a range of different clades and in many different species that have acquired 2A in this conserved position amongst all these different lineages of non-LTR retrotransposon, but they've lost ORF1. And the next major class of genes that we found cellular 2As in was in invertebrate nod-like receptor proteins, or NLRs. So these things are cytoplasmic proteins, and here's a leucine-rich repeat domain that senses the presence of PAMPs or DAMPs within the cytoplasm. Here's a ATP-dependent oligomerization domain, and here's the death domain, PYR, that signals the presence of these DAMPs or PAMPs in the cytoplasm. And what we find in this case is that these genes in these invertebrates, or some of these genes, have acquired a 2A-like sequence at the beginning of the open reading frame. And this had us scratching our heads as to why you'd want to have 2A at the beginning of an open reading frame. And it turned out that these 2As possess both the translational recoding activity and they can function as signal sequences. So the idea here is that you've got partial levels of cleavage or recoding, such that if the 2A does not recode itself away from this, it targets that to the exocytic pathway. If 2A does recode itself away, then presumably that ends up in the cytoplasm of these organisms. So that's been an interesting recent departure for us on cellular 2As. But getting back on track, viral 2As. Up until now, I've been talking exclusively about foot and mouth 2A, but, and this is one for picornavirus anarax or ICTV aficionados, what I've done here is I've taken one sequence of the polymerase from each of these, I think it's 48 genera now within the picornaviruses. And down here, we've got the enterovirus genus, but I've split it up into species. And if you align these polymerase sequences and generate this kind of phylogenetic tree, and then superimpose upon that tree the way that they separate their capsid protein domain from the replication protein domains, I think you'll agree there are two lineages whereby that is accomplished by that 3C proteinase in the P3 region. You've got a lineage here, including 1, 2, 3, 4, 5, etc., a number of genera, different genera within the picornaviruses that use this ribosome skipping mechanism. And then in only one genus, the enteroviruses, you have this 2A proteinase. So it's not just foot and mouth disease virus, it's a range of different viruses within the family Picornaviridae. But what's more, if you zoom out, so here we've got picornaviruses, and now we're looking at the alignment of the polymerase domains with insect viruses, dicystroviruses, and ifloviruses. What you can see that there are lineages of these viruses that also use 2A to separate their capsid proteins away from their replication proteins. These are viruses here have been implicated in colony collapse disorder, Israeli acute paralysis virus, acute bee paralysis virus, cricket paralysis virus, etc. So we're now building up quite a database of 2A-like sequences. You even get them in double-strand RNA viruses, for example, human porcine type C rotaviruses. It appears here that these viruses have used 2A to graft on an RNA binding domain at the end of segment 3 in comparison with these type A and type B rotaviruses. Insect cyperviruses and totiviruses within crustaceans. So it's not even just picornaviruses, it's a range of different positive strand RNA viruses and some double strand RNA viruses too. So the way that all the 2As I've been talking about are active, and the way that we can determine that is just use this simple reporter system that I mentioned before, dial in or test 2A sequence, analyze these things in these cell-free translation systems. And this is just a very small proportion of the 2A-like sequences that we've identified and shown to be active. But now, what we're doing is building up a repertoire of 2As that we can use for biotechnology and biomedicine. So in terms of eukaryotic protein expression, the problem has always been is how to get coordinated expression within the same cell. So if you do a mixed plasmid co-transfection, gene 1 and gene 2, on separate plasmids, you may get good co-expression, but that is across the whole population of cells on the cell sheet. And only a very small number of individual cells will co-express both your proteins at similar levels. This is probably what you're wanting from that kind of experiment. So to try and solve that problem, people use these kind of arrangements here with a promoter, gene 1, tandem promoter, and gene 2, or divergent promoters. And these approaches didn't really provide a substantial advance over just the simple mixed co-transfections. Now, co-expression in prokaryotes is not a problem. And the reason why it's not a problem is because they initiate their translation from these internal ribosome binding sites, or Shine-Dalgarno sequences. Here's the initiation codon here. So there's no involvement of the 5' end at all in initiation in prokaryotes. Ribosomes bind to these internal sequences, which means that mRNAs can be polycistronic. So they can organize genes into operons and coordinate protein expression just via the inclusion of ribosome binding sites in the intergenic regions. However, in eukaryotes, that you can't do that because initiation occurs at this 7-methyl G-cap structure here. So there's only one site of ribosome binding. There was a partial solution, again arising from picornaviruses, to this problem, which was to have bicistronic mRNAs, whereby you'd got translation of the first open reading frame occurring in a cap-dependent manner. Then you had an intergenic region comprising an IRES, which could then drive translation in a cap-independent manner of the second gene in that bicistronic mRNA. The problem was that if you looked at the stoichiometry of the products, you got about 10-fold less of that than of that. And the IRESs showed variable co-expression between different cell types. And I think you can figure out where we're going with this, is that the solution now was to use 2A. So you can concatenate open reading frames into a single open reading frame, a single transgene, using multiple 2As. All one has to do is to remove the stop codon at the end of these genes so you create a single open reading frame. And when that's translated, you get protein 1 to a protein 2 initiates with a proline, 2A as a C-terminal extension, etc. We and others have looked at this quite carefully, and unlike the in vitro translation systems I mentioned before, when you use this strategy to co-express stuff in healthy cells, what you observe is a 1:1:1:1 molar ratio of the products. In an early experiment we did to try and confirm this notion of 2A acting within the exit tunnel of the ribosome and to show its utility as a co-expression system, we created this construct here. Yellow fluorescent protein, bizarrely shown in red, cyan fluorescent protein, bizarrely shown in green. But the idea was here that we incorporated what was formerly an N-terminal signal sequence at an internal site here. So the argument ran that if 2A did genuinely work co-translationally, you would reveal this as a nascent N-terminal feature, which could be recognized by signal recognition particle and potentially target that to the endoplasmic reticulum. And that's exactly what happened. The first protein uniformly distributed throughout the cytoplasm, diffusing into the nucleus, whereas the second one is definitely targeted to the endoplasmic reticulum. We've got another 2 here in PAC. We just did that for laughs so that we could use the puromycin, the PAC resistance, to establish cell lines more easily. So, what's this been used for in the real world? Well, when thinking about high-value proteins, some work that we did with the late Bob Collins from the Institute for Animal Health at Compton was to co-express the P40 and the P35 subunits of IL-12. This was extraordinarily useful. It was done for bovine, ovine, and porcine IL-12s. Monoclonal antibodies, for example. Here, you've got the heavy chain and the light chain, but what these guys did was to introduce a furin cleavage site between the heavy chain and 2A, such that when this transited through the Golgi apparatus, the furin present in the Golgi actually trimmed off 2A from the heavy chain. Both signal peptide sequences are recognized by SRP, both are removed by signal peptide peptidase. So the point here is that not only can you get co-expression, but you can target things to different sites within the cell as well. And then you can use it, for example, to actually concatenate genes of biochemical pathways. So here, for example, we have a whole operon, as it were, created using different 2As together with this gene here, and that actually now produces constitutively auto-bioluminescent human cells. And this was just comparing different promoters here. But so you can actually now have a single transgene putting in this entire pathway. And one of my particular favorites was this work done here, where 2 pathways were concatenated together, one to do with beta-carotene synthesis, and one via lysine. And when you co-express that, all of these proteins in Pichia pastoris, it turned purple because you'd got the 2 pathways generating an orange and a purple product within that Pichia. Macromolecular complexes, for example. So a guy called Dario Vignali at St. Jude's Hospital in Memphis, he used, again, different 2As to create one lentivirus encoding that artificial polyprotein and another lentivirus encoding the A and B chains of the T cell receptor. When these were put into mice, they got construction of a novel T cell receptor complex in those transgenic mice. So you could use this to make whole macromolecular complexes. Transgenic plants. So this is the work that my wife Claire did when she was at AstraZeneca. The idea here was to try and co-express cat and gus using 2A, and what you can see is nice expression, co-expression of these two things in transgenic tobacco lines. And Neil mentioned vitamin A deficiency. I won't read through all this, but as you know, vitamin A deficiency is a big, big problem in many areas of the developing world. And there was a big EU-funded project by Peter Bayer and Petrikis to try and convert geranyl pyrophosphate into beta-carotene. And that beta-carotene then could be converted in our bodies into vitamin A. So they identified that in rice, the last precursor of beta-carotene that could be used was geranyl pyrophosphate. They had to make transgenic rice expressing 4 proteins to get from geranyl pyrophosphate up to beta-carotene. And the way they did that was through reiterative transgenesis of the rice, which took quite a long time, introducing transgenes one after another. However, these guys, what they've done is actually got 2 bifunctional proteins and linked them together either via a 2A or co-express them via an IRAS. They wanted to compare these 2 different strategies to co-express these 2 bifunctional proteins. And that's the data. Here is rice now turning yellow because of the beta-carotene that you're making, and that's them linked via 2A, and that's them linked via the IRAS. And you can see that's the wild-type rice, that's the first generation golden rice, and that's the second generation golden rice produced using this kind of co-expression strategy using 2A. So 2 of the world's most common transgenes are glyphosate tolerance and the expression of Bacillus thuringiensis toxins, which can stop pest predation on these crops. And you can now stack these 2 transgenes into a single transgene via 2A. Here's one Bt toxin, glyphosate tolerance, and you can stack and perform this transgenesis in using a single transgene. And here it shows that the wild type being eaten by this pest, the transgenic is resistant. Here's the transgenic resistant to glyphosate, whereas the wild type is killed by glyphosate. So that's another powerful aspect of 2A, is that you can make your transgenesis much more efficient by concatenating your transgenes into a single transgene, and that ensures coordinated expression. Transgenic animals. So again, this is one that I've cherry-picked from the literature. This is transgenic mice now expressing tomato targeted to membranes or GFP targeted to the nucleus. And here you can see these beautiful images of different tissues from that transgenic mouse. Transgenic zebrafish, the same thing, GFP and M-cherry, and you can see this fantastic co-expression within the zebrafish. And this is quite an interesting experiment done in pigs, I think in Korea, whereby they wanted to make yellow and cyan and tomato and green. So that's the construct that they used, all linked via 2As. And that's the transgenic pig you get out from that that's fluorescing in these multiple different colors. And then, perhaps on a slightly more serious note, was the adoption of the use of 2A for cancer gene therapies. Now, I mentioned about the work of Dario Vignali constructing a T-cell macromolecular complex in transgenic mice, and that included the A and the B chain of the T-cell receptor. And some work, again done in the States, was to address the problem of melanoma. A classic syndrome here are Scots living in Australia, developing metastatic melanoma. And it turned out that somebody in America had got metastatic melanoma and the tumor had spontaneously regressed. And they managed to track down the antigen that was responsible for that and how that antigen was being recognized by the immune system. And it turned out this individual had got T-cell receptor A and B chains that were capable of recognizing the MARC1 antigen on these metastatic melanoma cells. So the idea was, I now contract metastatic melanoma. You take the T cells out of me, transform them with the A and the B chain from this particular person who mounted a protective immune response, then put these autologous cells back into me. And it works. So this is the example I'm showing you for metastatic melanoma, but there's a whole range of different cancers now that are being treated in this way. And as you can see, these subcutaneous lesions disappearing, these tumors regressing or disappearing. And I must say, I got a real buzz when I saw that paper coming out. So this strategy is now being used in a whole range of cancer gene therapies. And then regenerative medicine, again, I live vicariously through these kind of publications, but the problem here is that the sources of, I mean, people neglected to save my umbilical cord when I was born, heaven only knows why, but getting sources of adult stem cells turns out to be pretty tricky. And even if you can get them, maintaining them in tissue culture is nigh on impossible. So the idea was, can one make stem cells from differentiated tissue? And Yamanaka, who got the Nobel Prize for this piece of work, he transduced cells with combinations of lentiviral constructs, each one encoding an individual gene. And by this pooling of constructs and transforming of cells, he identified these 4 proteins, transcription factors here, which could knock a mature differentiated cell back into an induced pluripotent stem cell state. And this is just kind of like absolutely exploded now in the whole field of regenerative medicine. So I think you can probably all guess where this is leading to. You need to co-express these 4 transcription factors within the same cell. And this is actually a diagram from work done by a group in Edinburgh, CMYC, and it works astonishingly well. So this has now become the industry standard, I think, for making induced pluripotent stem cells. And just to flag up that these are 2As that we identified from these other viral genomes. So I think that's what we brought to the party, was the ability to use a whole range of different 2As in these constructs. And 2A has been used in a whole range of genome editing technologies, in this new technology of chimeric antigen CAR receptor technologies, optogenetic control systems, and I like to keep track on who uses these things because it helps with grant applications, and it's hubris as well. But so there are now around about 700 patents that cite the use of 2A. It's being used in many biotechnological products, actually some by the demonstrators we had outside. And if you look on, as I say, we keep track of who uses it, and now there are over 1,000 papers that refer to our papers on 2A, citing the use of 2A. So finally, I would like to extend my thanks to Unilever for actually making this prize possible, for the Society for putting on, I think, what is an absolutely fantastic meeting and awarding the prize to me, but I think it reflects on people in the lab. And then finally to you guys for coming along. Thank you very much.